Right, I'll get started. So welcome everyone. Good morning to those of you in the States and afternoon to those of you in Europe like myself. This is Wing, the Congress Director for the World Vaccine Congresses, and I'm delighted that you can tune in today. I hope that you can stay with us throughout the second COVID-19 series put together, and that's obviously today, Wednesday, and on to Thursday as well, all at this time, 11 a.m. EST, with speakers, of course, from Inovio, as well as Novavax and Moderna. I would also like to take this opportunity to thank our webinar sponsor, Sino Biological, for supporting and helping us deliver today's webinar with Inovio. And just a gentle reminder, especially if you haven't tuned in with us before last month, and you can all submit questions using the control panel, and we will do our best to bring these up during the Q&As after the presentation itself. And these sessions are also video recorded, and you will get a link to it tomorrow. And I'm sure we'll be saying this message again afterwards. So now I finally have the pleasure to introduce back our wonderful moderator, Dr. Philip Krause, Deputy Director, OVRR CBER at the FDA. And as his title suggests, he oversees activities associated with the regulatory review of products regulated by the FDA's Office of Vaccines Research and Review. He also contributes to the global public health as an ad hoc advisor to the WHO, and most recently, development of the research roadmap for the vaccines against SARS-CoV-2. So without further ado, I'll pass over to you, Phil, to introduce the session, and Kate.
Thanks a lot, Wing. Today we really have a great opportunity to hear from Dr. Kate Broderick, who's the Senior Vice President for R&D Preclinical at Inovio. And Inovio is a really exciting place to be right now because they're one of the companies that's deep in the hunt for a COVID-19 vaccine. As many of you know, their primary technology is DNA vaccines. And Kate has a broad background really in doing work that has facilitated this, especially in the area of developing electroporation devices that aid in delivering DNA vaccines. She's a well-respected scientist and is really a major part of Inovio's team that has enabled the company to be as successful as it has. And her talk title today will be Advantages of DNA Vaccines in Response to COVID-19 and Inovio's Progress. So we look forward to hearing your talk, Kate.
So yeah, good morning, everybody. My name's Kate Broderick. I'm representing Inovio Pharmaceuticals today. I'm going to be talking about our activities on, of course, the COVID-19 efforts that we've been working on over the last few months, kind of giving you an update in regards to where we are today, where we've been over the last few months, and where we hope to be in the future. So just a quick presentation outline, going over a little bit, I think, about Inovio for those maybe who aren't familiar with us, a little bit about our DNA medicine platform, the applicability we believe for emerging infectious diseases and why that puts us in a good position to respond to the outbreak that the world is dealing with at the moment. A little bit of an introduction to COVID-19, not that I think anybody's going to need it. Talk a little bit about our MERS data, which I think provides a very nice foundation to what we're doing today. Talk about our preclinical data and the challenge data that we're currently developing, our phase 1 progress so far, and then where we hope to go in the future. So before we get to the basics about DNA medicines, about DNA vaccines, just a little bit about Inovio. So we are, our headquarters are based in Plymouth Meeting in Philadelphia. I'm based in San Diego. That's our research hub. We have all R&D activities here in San Diego and also all of our manufacturing activities related to the device. We are about 200 people currently, so a mid-sized biotech, I guess. And although, of course, I'm going to talk primarily about COVID-19 today, we also, I just wanted to remind everybody, we have 2 concurrent phase 3s running at the moment for our lead program, which is our therapeutic HPV vaccine, and also a very interesting phase 2 for our GBM program, where we're going to be announcing the data of that extremely exciting trial in the next few days. We also have a broad portfolio in the infectious diseases space, and I'll touch on some of that as we go through the talk today. So coming into some of the key characteristics of the technology that Inovio has developed over the years, specifically pertaining to our DNA medicines, DNA vaccines. So let's start in the center of this graphic, and that's really the central tenet to the basis of our technology. So that's the marriage of our highly optimized DNA plasmids with our smart delivery technology. And both of those aspects are being used for the COVID-19 program that I'm going to be discussing moving forward, but of course are also central to all of the clinical programs that we've developed over the years at Inovio. And one, I think, of the most important characteristics of our DNA vaccines is the ability to produce really robust, balanced immune responses. So by that I mean not just very robust humoral responses, but also T-cell responses too. And obviously I'm going to talk about that in the context of COVID-19 as we move through that. There are some inherent, of course, characteristics to DNA vaccines. A popular myth is on the integration side, but of course DNA does not integrate into the host genome. There's also the ability to boost. There's no vector immunity, so we can also very efficiently boost, of course. And also by the nature of the formulation of DNA plasmids themselves, we can have cocktail vaccines or multi-antigen vaccines into a single vial. And so preclinically at the moment, we're looking at combining our vaccines for hemorrhagic fever, so Ebola, Lassa, and Marburg together, and the same for mosquito-borne diseases. So you can see from the perspective of the patient getting a single shot, but being vaccinated against multiple targets at one time. Another aspect which I think is particularly pertinent in today's discussion is the rapidity and the scalability of our manufacture. And that's certainly something I can talk about more in detail at the Q&A session, but that's very important. Here in San Diego, we have our preclinical DNA manufacturing labs where we use benchtop bioreactors to produce small batches of our vaccines for our animal studies. But of course, when we're talking about our GMP material, that gets scaled up very, very rapidly up to reactors, the size of rooms, for instance, just to give you an example. And last but not least, I think a key differentiator for us is the stability profile of our DNA. It's long-term storage in refrigerated 2 to 8 conditions, so we don't require a frozen cold chain. But I think importantly, specifically as we think about the deployment of our vaccine candidates in less developed settings and also under kind of critical circumstances like we are experiencing at the moment, we can have no loss in potency of our vaccines at room temperature for over a year. And actually at 37 for approximately 2 months. So that's really quite a nice characteristic, I think, to have, especially as we're thinking, of deploying vaccines and resources where access to stable cold chain might not always be confirmed. And then last but not least is the ability to rapidly design our vaccines. And I'm obviously going to talk about that in depth because that really specifically relates, of course, to our COVID-19 efforts. So moving on, so what specifically about our platform technology makes us a good fit for emerging infectious diseases? And obviously I'm going to use COVID-19 as a working example here. Well, speed, as I just mentioned, is a key player here. As some of you may or may not know, It took us 3 hours from the receipt of the SARS-CoV-2 sequence from the Chinese authorities to having a designed DNA-based COVID-19 vaccine. And I'll elaborate a little bit on what that was. So really the design and manufacture of this particular DNA vaccine represents really a plug and play process in which we insert the target antigen sequence into a highly characterized and clinically tested plasmid vector. And this process has been used extensively, not just here for COVID-19, but also, of course, for our other infectious disease targets such as MERS, Ebola, Zika, the flu, etc., etc. As I mentioned in the previous slide, rapidity of manufacture. So we designed the vaccine itself in 3 hours. We immediately had that synthesized and put that the next day into small scale manufacture so that we could move very, very rapidly into our lab-based testing for in vitro and in vivo readouts. And also, I guess, falling under that speed bullet point is our ability to harness established regulatory platforms. You know, we have run a significant number of clinical trials now, and the FDA has reviewed our processes many times, and so is very familiar with our technology. And as such, we don't need to run toxicology studies for our new products, which of course saves us a lot of time, especially in a situation like we're in today. So the second bullet point is our safety profile. We've now treated over 2,000 subjects in the clinic with our DNA and our delivery technology, which amounts to over 6,000 doses. And we really are very comfortable with the profile that we see in the clinic. And I can elaborate on what we've seen so far for our phase 1 trial for COVID-19. So breadth of immune responses. So as I touched on earlier, and I'll elaborate on in further slides, we're able to generate broad and robust B and T cell responses, and of course the all-important neutralizing antibodies, and I'll give you examples of that for our COVID-19 vaccine. Also, another key point I would like to mention is the ability of our platform to generate immune responses in an elderly population, and this was specifically a trial that we carried out a few years back in an elderly population using our flu vaccine, and it was really very heartening for us to see that we were able to generate T-cell responses in that notoriously difficult population, and we have every hope that that will be applicable here for our COVID-19 vaccine as well. So duration of response is a very key component here, and certainly with our lead product, our VGX-3100 therapeutic HPV product, we've seen immune responses over 3 years post immunization. Of course, we haven't had the luxury of looking at the duration of responses for our COVID-19 product yet, but we believe that they would fit in the same bucket as we've seen for our previous other products. We've also, for one of our other hemorrhagic fever products, demonstrated protection 12 months following last immunization. So a really nice demonstration of protective durability of our immune responses as well. So in the efficacy bucket, of course, the key component here, we were the first group to demonstrate clinical efficacy of a DNA vaccine combined with our delivery technology. That was in our phase 2 for our HPV program. And we also, of course, have multiple published reports of protection from challenge in animal models, and that's for MERS, Lassa influenza, Ebola, and Zika. And I'll touch on our MERS data in the following slides. And last but not least, of course, again, is our stability of our product. I do think that's a key component for us. It does not require frozen storage and really has a very desirable stability profile at both room temperature and higher temperatures as well. So as I mentioned earlier, I don't think anybody on this call really requires any background on COVID-19. So I included the slide not because I felt you needed to know any background, But just because when I was putting these slides together, I found this particularly compelling, because I used this slide first back in March, and I thought, well, I'll just recycle it again today. But what really struck me was the differences in the confirmed cases, and unfortunately, of course, the number of deaths between March and we're in May today. If you could compare the left and right panels, in March we were talking about just over 100,000 confirmed cases. Today we're talking over 3.5 million confirmed cases. And of course, unfortunately, the deaths have tracked with those numbers too. So it's an incredibly fast-moving and devastating disease that we're all working together on at the moment. I would draw attention on the right-hand side to this particularly stunning heat map of outbreaks of the disease in the US. It's quite a sobering picture, especially as I read today about continuing outbreaks in the Midwest, where currently this picture looks fairly clear. Unfortunately, it looks like there's some hotspots springing up there too. The mortality rate certainly varies, as I'm sure you all know, country to country, area to area. We're certainly seeing differences there. It's somewhere in between the 1 to 2.5% mortality rate. But really something to focus on is the high morbidity rate that lasts somewhere from weeks to potentially months to potentially years. I don't think we fully know the impact of this disease yet. So our strategy at Inovio for our COVID program is really simple. So let me move through it, because I think it sets a nice foundation for the slides that I'll cover as I move through my talk. We set ourselves the goal to move rapidly into phase 1 human testing to demonstrate safety, tolerability, and immunogenicity. And you see we've got a big check mark there, and I'll talk more about where we are in that space as we move through the slides. We also took it upon ourselves to generate very early on a very strong preclinical package of data supporting our candidate, and I'll talk about that data too. And of course, we're in the throes at the moment of generating the supportive animal efficacy challenge data, and I'll talk about that as well. We would like to, of course, on the back of the phase 1 data, move as fast as possible into human proof of concept efficacy studies, and I'll talk about our plans for that. And we, of course, we want to leverage external funding to support this effort, and I'll talk about how grateful we are to our funders and supporters that have backed us thus far. We're obviously also expanding our manufacturing capabilities as we think about deploying this vaccine. The need to scale up is very, very key, and we're certainly very, very focused on getting into the hundreds of millions of doses as soon as possible. And of course, we're looking to expand our global partnerships. This is already for us a global effort, but we're already partnered with 2 teams, one in Korea, one in China, to run parallel phase 1 trials as well. So as I mentioned, we're extremely grateful to our supporters and partners who have assisted us getting where we are today. CEPI, I would say, has been there with us really from day one, and we're extremely grateful to their ongoing support. Also, the Bill and Melinda Gates Foundation have been historical supporters of us and continue to support us for this program too. As I mentioned, we'll be running a concurrent phase 1 trial with IVI in Korea, and also a phase 1 trial with Advaccine in China, and we're very grateful for their partnership. On the animal challenge side, we're extremely grateful for the partnerships at CISRO, Public Health England, and Peking University, who are supporting our challenge efforts. We've also received funding from the Department of Defense and the Defense Threat Reduction Agency here in the US. and our ongoing partnership with the Wistar Institute and with the University of Pennsylvania, all of whom we're extremely grateful for their ongoing support, and it really helps us come together and find a solution for this devastating disease as fast as possible. So our response to COVID-19 outbreak really builds upon our prior experience in developing a MERS vaccine, and I'm going to touch on that because I really do think that's key to us moving forward. So immediately upon really understanding the threat that initiated in China, we employed our synthetic DNA vaccine technology to develop a vaccine against COVID-19. As I mentioned, it's particularly amenable to this type of outbreak setting, as we're able to really accelerate our candidate discovery timelines, rapidly design multiple and test multiple candidates, manufacture large quantities of the drug product, and leverage our regulatory precedents to support rapid entry into the clinic. We designed our candidate based on our experience with our MERS protein, and that was using the spike protein design, and I'll go into details about that moving forward. So talking a little bit about our MERS program, so this was called INO 4700, and abstract of some of the new and exciting research based on this particular program is going to be presented this week at ASGT. But this is just a very high level summary of where we are. So this was our MERS vaccine candidate, which we really got into gear working on following the outbreaks in the Middle East and also, of course, in Korea, and started doing some extensive preclinical work in this area. And we were extremely pleased to see that we were able to demonstrate 100% protection from clinical disease in primates after 2 immunizations using our intradermal delivery. And actually, really quite remarkable, 75% protection after a single immunization. So we're really very emboldened by that data, very supportive. We also investigated immunogenicity in a variety of different models for our MERS candidate, including, of course, primates, which were the model for the challenge study, but also mice and indeed actually camels as well, and that's a story in itself. But we were able to really show strong cellular and humoral responses after both 1 and 2 doses of our vaccine. So we're very excited about that. And really the preclinical data was the foundation, of course, for us to move on very quickly into our clinical testing for this vaccine. And this has been supported by our partner CEPI, and we're very grateful for their support. So we first carried out a phase 1 trial in the US and then a phase 2 trial in Korea. And remarkably, in the Korean study, after a single immunization, we saw 76% seroconversion and 80% seroconversion after 2 immunizations. And again, like we did in the preclinical models, we saw strong and broad cellular responses noted at all time points. So we really felt that this gave us a particularly strong foundation to use the same design, the same strategy for designing our SARS-CoV-2 product, which I'll speak about now. So this is really rather a busy slide, but I think it maybe pictorially represents what our working lives have been like for the last 4 months or so. It's been quite a whirlwind, as you can imagine, really from receiving the viral sequence to immediately getting that design and moving it into laboratory testing and towards the clinic. So what you can really see here is our preclinical work where we've tested in a variety of different animal models, starting with mice, guinea pigs, rabbits, all the way up to primates. and are now kicking off those all-important animal challenge studies in ferrets, mice, and primates as well. We submitted this package, this initial package, I should say, for our small animal studies to Nature Communications, and we're really very excited to announce that this week that's been accepted for publication. So the full paper should be available in the next few weeks as a full print version. As part of this process, we of course designed a lot of new assays. We looked specifically at ACE2 spike blocking, biodistribution of IgGs in the lung, which of course is key for prevention of lower respiratory disease. And we also were able to get important both pseudovirus neutralization data and live viral neutralization data with our partners. And that's really built up to us being able to say that we treated our first patient in our phase 1 trial on April 6th. So, a remarkable 83 days from receiving the sequence to treating our first patient. So it's certainly been a very busy, challenging time, but something that we're really very proud of, the progress that we've made so far. So going right back to the start, to how this all started. So as I mentioned in the opening comments, really we put into play the technology that we've been building up over many years now, including our gene optimization algorithm. And as I mentioned, really the design and manufacture of this COVID-19 DNA vaccine represents really a plug and play process in which we insert the target antigen, in this case, of course, the spike protein, into our highly characterized and clinically tested plasmid vector backbone. And really, we have a huge amount of experience doing this in the past with our other infectious disease targets and also our other non-infectious disease targets as well. So moving through, the sequence, of course, was published, of which we're extremely grateful to the Chinese authorities for their speed in that matter. We uploaded that into our algorithm. 3 hours later, we were given a design of that was synthesized and then cloned into our expression vector. Then, of course, we used standard expression, where we looked at expression in a variety of different cells in culture, and then used a Western blot, of course, to confirm that the protein was indeed what we expected it to. Of course, it's the full-length spike protein that we're using here, and that's really a commonality from our MERS program, where we, of course, were able to show really very impressive protection and also excellent clinical data as well. So then, as I mentioned, we immediately went into our preclinical testing phase, starting with mice. And the data I'm showing you here is after a single dose of INO 4800, so our COVID-19 DNA-based vaccine. And one thing that we were able to show here is really the rapidity of the responses that we were able to see. In the case of the mice work, we used a variety of doses just to really find what was the best dose for this particular model. So we used 2.5 micrograms or 10 micrograms. And you can see in this top panel for our interferon gamma ELISpot readout that we were able to detect really quite robust T-cell responses and broad T-cell responses at day 7. And I do think that rapid cellular response could have potentially to lower those viral loads and could potentially reduce the spread of SARS-CoV-2 and COVID-19 illness if that translates to humans. We also, of course, were very interested in looking at the humoral responses, and you can see here we were able to demonstrate very robust humoral responses. This is to the S protein, but we also, as I'm sure many other labs around the world, have looked at a variety of different binding proteins to show binding titers as well. So from our work in mice, we moved over to guinea pigs, and just to give the background on guinea pigs, they are an extremely good surrogate model for human skin. And as you may know, we are using intradermal delivery for our SARS-CoV-2 vaccine. So the guinea pig is really a very nice model for us to use our clinically slanted delivery approach in an in an animal model. So again, as we did with the mice, of course, we looked for T cells and humoral responses, and again we saw very nice, robust B and T cell responses. And the ELISpot was actually designed by our development team here in house for us to be able to track T cell responses in guinea pigs. But importantly, I'd also like to bring your attention to the ACE spike blocking data that you can see in the middle of the figure here. The induction of antibodies capable of inhibiting spike protein engagement of HOPE receptors is considered, of course, relevant for SARS-CoV vaccine development. So therefore, we examined the receptor inhibiting functionality of INO 4800 induced antibody responses and developed an ELISA-based ACE2 inhibition assay as a surrogate for neutralization, and really saw very, very nice blocking of the receptor there in our immunized animals. As I mentioned, we also looked for IgG biodistribution in BAL, and were able to detect really robust titers in the lung, which of course, as I mentioned, is something that we believe will help protect against the spread of the virus itself as a mechanism. So although I'm talking about this in kind of a sequential scenario, actually, of course, we did this for rapidity's sake in parallel. So we started working on primates very, very quickly, as of course we wanted to use them in our challenge studies, so we immunized them very quickly. We have two groups. One is 1 mg dose of our vaccine, and the second is 2 mg dose, and that is a reflection of our clinical program as well. And again, just like we saw in the mice and guinea pigs, we were very, very encouraged to see the rapidity of the responses that we saw both on the T cell and B cell assays, the rapidity and the robustness of those responses. And you can see on the T cell side of things that we get really a very nice broad distribution of our peptide pools there, which is very important as we think about specifically responses against the spike protein. So the all-important, of course, neutralizing antibodies. We ourselves in-house developed a pseudovirus assay, which we have used to assay our animal studies, but we were also very happy to be able to collaborate with our partners at the National Laboratories of China at Peking University for our mouse live neutralizing antibodies, and also with our fantastic partners at Public Health England, Porton Down, who carried out our guinea pig live neutralizing antibodies. And I think you can see both for the mouse panel and the guinea pig panel, we were really emboldened by the levels of neutralizing antibodies that we were able to see both in our live and our pseudo neutralization tests, and that's all being published in the Nature publication. So very key, of course, the immunogenicity data is of course vital and build a very nice robust data set, but really key, of course, is the demonstration of preclinical efficacy. So we are carrying out a mouse challenge with again with the National Laboratory in China through our collaborators at AdVaccine. And we're also performing a ferret challenge study with our partners at Cicero in Australia. And last but not least, we have just initiated our primate challenge with our collaborators at Public Health England at Porton Down. So we're really very excited to be working towards generating this vital preclinical efficacy data. So using our preclinical data that we generated, we were very keen to move into the clinic as soon as possible. As I mentioned in my early comments, we were able to achieve that in 83 days, which certainly is a record for us. We began our study on April 6th, and due to the overwhelming interest that we received in this study, we were able to fully enroll our 40 subjects by April 27th, and I'll touch a little bit on more details of that on the next slide. We, of course, hoping that the phase 1 data looks good, which we have every confidence that it will. We want to move very, very rapidly into a phase 2/3 study, and we're hoping to get that up and running in the summer timeframe. As I mentioned, we're also running in parallel 2 ex-US studies, one in China and one in Korea. And those have been collaborations with Advaccine in China and IVI in Korea for our joint clinical development. So a little bit more details about our phase 1 progress. Firstly, this was done in partnership with our collaborators at the University of Pennsylvania and also our clinical research labs in Kansas City, both of whom we have worked extensively with in the past for clinical trials, so are very, very strong proponents of our technology and have been really fantastic collaborators and supporters. So it was a real pleasure to be able to work with them on such a critical clinical program as this. So this was a small sample size, 40 patients, and it's really to give us preliminary assessment of safety, tolerability, and of course immunogenicity. And we were able to use our historical experience with our clinical trial sites to quickly start up those activities, get everything in place and ramp up to start our trial on the 6th of April. As I mentioned, we were able to enroll incredibly quickly, and that's absolutely 100% due to the incredibly amazing, motivated study sites and also our volunteers of whom we, without their generosity to volunteer their time to these studies, we would not be able to be in the position we're in today. It was really very heartening to listen to some of the stories of the volunteers of our clinical trial, some of who knew people, unfortunately, who had actually died from COVID-19, and that was really very motivational for them to want to be a volunteer in our study, and we're incredibly, incredibly grateful for their participation in our study. And the picture there on the upper right hand is one of our subjects at our Kansas City site. So our phase 2 development plans, we're, as I mentioned, of course, moving on with our phase 1 trial, which is ongoing. We're also looking to perform a phase 1 study expansion, and we're in discussions about that possibility at the moment. And that's really to look at responses in 51 and over population, which is of course going to be very key as we think about rolling this vaccine out, and also looking at potentially dose sparing regimes, so looking at potentially lower doses for our vaccine as well. Then, of course, as you can imagine, we've been thinking a lot about what an efficacy trial will look like. At the moment, we're kind of coalescing around a large phase 2/3 trial, specifically targeting high-risk occupations, so those would be healthcare workers, support staff, etc., for our targets for this trial. And that's really ongoing, and we're working very diligently to try and get all those packages in place to support those activities and moving as fast as possible into those studies. So our major accomplishments to date, really this is just kind of a list to kind of assess where we've been over the last few months, and with a mind to where we're going. So as I mentioned, obviously, immediately we designed our DNA plasmid based on the native spike protein sequence. We immediately pulled the trigger on animal immunogenicity data, and that was both in small and large animals. And of course, in the large animals, we're able to deliver the full human dose. And really, from the basis of that study, we were able to detect neutralizing antibodies, which was very, very key for us. As I mentioned, our animal challenge model studies are underway in mice, ferrets and primates, and our first publication's now been accepted to Nature Communications. We're of course interacting frequently with the FDA in regards to our ongoing phase 1, but also in regards to starting our phase 2 interactions. And of course, as you can imagine, very key at the moment is the scale up of our GMP manufacturing of our investigational product. We're very, very focused at the moment on getting into the hundreds of millions of dose number, and we're working with our existing collaborators and also new partners to ensure that we're able to rapidly and scalably get those dose numbers up. Of course, as you can imagine, since we're in clinical testing now, our analytical team has been developing the appropriate analytical assays for our human trials. And our first patient was dosed on April 6th, which is a huge milestone here for the team at Inovio after, really remarkable, really determination of the team, and I can't thank them enough for their hard work and their dedication to getting us to that point. We enrolled the trial fully on April 27th, so now all of our subjects have received their first dose, some into their second, and of course we're planning for our clinical studies to commence in China and Korea in the near future. And discussions are ongoing with additional CMOs, as I mentioned, to scale up manufacturing of our plasmid so that we can really get into those hundreds of millions of dose numbers as soon as possible. And that just really leaves me to thank you, the audience, for listening. I really appreciate your time today. And of course, I'm very, very happy to answer any questions that you might have on the presentation. Thank you so much.
Thanks a lot, Kate. This is Phil. There are indeed a lot of questions that people have brought up. And perhaps a good way to start is with the basic technology of DNA vaccines. The wrap on DNA vaccines, which of course have been around for a long time, is that they've generally worked very well in animals, but have been disappointing in humans. And I know that Inovio has done a lot of things to increase the responses to DNA vaccines in humans. And some of these you described fairly perfunctorily. But maybe you want to say a few words about what are the strategies that you've used in order to make DNA vaccines work better in humans.
Yeah, absolutely, Phil. Thanks. And certainly many, many years ago, when DNA vaccines kind of first burst onto the scene and had a lot of kind of potential behind them, when they initially went into humans as just a naked DNA injection, they were indeed quite disappointing data. And many people left the field at that stage, but Inovio and many others, of course, didn't, and went back to the drawing board and really said, what is it that we're missing here? And I think very, very clearly, certainly to us at Inovio, what we were missing back 20 plus years ago was really the delivery technology to ensure that the DNA was getting efficiently into the cells. And that's really, I think, where Inovio's taken really quite a leadership position in the DNA vaccine field and coming up with really very enhanced delivery technology, and that's of course what we're using in our clinical trials now. Really, that's absolutely key. So you might have the most immunogenic DNA plasmid known to man, but really without enhanced, optimized DNA platform technology, that's really not going to get you very far. So really, certainly in my opinion, it's that marriage of our smart delivery technology with our optimized plasmid that's really got us to the position where we're in today, where we really do see extremely robust and efficacious responses in human beings. And you know, I'm really very proud of where we are, you know, with our phase 3 with our lead product, and also just about to announce our data from our very exciting GBM, you know, a very difficult to treat cancer. But also in the infectious disease side, I think we've published now very extensively on our MERS candidates, of course, our Lassa fever, Ebola candidates, showing really that those results from 20 years ago are really very clearly a thing of the past. And with this advanced delivery technology, we're able to really truly see the potential of DNA vaccines.
So, and of course, you've played a big role in the development of the delivery devices. So congratulations on that. The selected device, of course, is an additional thing that you need with your vaccine that is not required with other vaccines. And we read in the newspaper that there's big concern even over finding enough of the glass to make all the vials that might be required for all of the doses of vaccine that need to be given. So can you tell us a little bit about how the device, the scalability of its manufacture, as well as the schemes for bringing the vaccine to the public all fit together in a way that you think will make a big difference in the pandemic?
Yeah, absolutely. And yeah, you're absolutely right. We do use additional delivery technology over above a single needle and syringe. But what we don't need is very elaborate cold chains. So we don't need -80 freezers and such like. So there definitely is a trade-off there. But certainly, questions about scale up are absolutely key and extremely valid in the situation that we're in today. As I mentioned during my slides, of course, we've partnered with several different entities on the DNA product side, but on the device side equally, although I didn't talk about it in the slides, we've partnered up. We have our own manufacturing capabilities here in San Diego. And we're also really speaking to other supply manufacturing technologies that would be able to scale up the delivery of the device. The one thing, if I could maybe just take an opportunity to speak about the device that we're positioning to use for our COVID-19, it's called our 3PSP technology. And for those who may not be familiar with it, it looks quite like an electric toothbrush. It's much smaller, I think, than people may have a sort of historical picture of what our delivery devices look like. This is a handheld device. It's very compact. It's very easily manufacturable. And so we're working with our partners, both our funding partners at the moment and other partners as well, to really ensure that we can scale up both on the drug side to those hundreds of millions of doses, but also to ensure that we have the appropriate number of devices to support that kind of rollout of those kind of numbers of vaccines.
A number of questions came through about the sequence of the spike protein that you're using, and in particular, you of course started with the Chinese sequence, but we're learning now that their European strains are circulating more. And there's a question also of whether you have pre-fusion stabilized your protein or whether there are other mutations that you want to talk about.
Yeah, absolutely. As you can imagine, I think, you know, us and really everybody who's working in this field is paying very close attention to the new strains that are circulating, looking out for any new mutations which, you know, could be troublesome or worrisome moving forward. So far, the mutations that have been publicly announced have no impact on our design. Coming back to the pre-post fusion question, we use the full length spike protein here, and really that's on the basis that that was exactly the same as we did for our MERS coronavirus, and we're able to generate robust neutralizing antibodies and C, of course, protection with that strategy. So we felt that was appropriate and well positioned to do that the same for our COVID-19, our INO 4800 design is what we did today.
So that would be a difference between your vaccine and many others, which include those prefusion mutations. Several people asked questions if you could just briefly contrast the DNA and RNA vaccine approaches. Both obviously involve nucleic acids. And what I'll say for those listening is, I see it's 11:56. The plan was to go to noon, but of course we got started late. So we're going to continue a little bit beyond noon to provide a chance for some more discussion and answering some questions. But I will break in right before noon to remind you of the talks, which are tomorrow and Thursday. But if you can go ahead and address the difference between DNA and RNA vaccine platforms, that would be great.
Yeah, absolutely. As you mentioned, Phil, quite rightly, they're both nucleic acid based platforms. And so both really allow us to that rapidity, I think, and has really come into play, particularly into the forefront here in the COVID-19 outbreak. The characteristics, I think the very positive characteristics of DNA vaccines in particular, I think is firstly, I think one of the very differentiating characteristics is the stability profile. You know, certainly DNA is a very stable molecule. RNA requires stabilization of the LNPs and then storage in a frozen temperature. But really, I think it's important as well that the differences in the way that the body responds really to DNA and RNA is really quite distinct. And we do think that is very key, I think, to some of the immune responses that we're able to generate with our DNA-based approach. DNA is not considered kind of an inflammatory molecule, so we do get a very strong Th1-based response from delivery of our vaccines. And so really we feel that the broadness of those responses and that both giving us humoral and T-cell responses is really very key in the DNA vaccine approach.
So I guess one other difference is that DNA includes CpG motifs, which may help to improve the immune response, which obviously mRNA vaccines don't. But your vaccine, as I understand it, is just the DNA injected without anything else added. Is that right? So it's relying on the internal structure of the DNA to also help induce that immune response.
Yeah, that's absolutely correct, Phil. Yes, and also the delivery technology itself acts in some way as an adjuvant to the DNA as well. So you've got the DNA component and then the actual delivery technology, which really is the key to bringing together those aspects and the way in which I believe that we're able to derive such a robust and broad immune responses.
Great. So several questions about what we've learned or what you've learned from your MERS vaccine. Obviously, that made it to a certain stage, did not find its way into phase 3. So people are asking why didn't that go farther? But they're also asking, what have you learned about, for instance, durability of immune response and perhaps the protection of the antibodies that are induced by the MERS vaccine because that has been tested in humans, right?
Yes, absolutely. So yeah, let me just recap a little bit on the MERS. So yeah, we did extensive preclinical work where we showed 100% efficacy of our MERS vaccine in a primate challenge. And maybe for those who are familiar with the MERS challenge, it's really quite a complex challenge. It uses multiple routes of inoculation for the animals. So I think seeing that 100% protection was really emboldening for us. We have now taken that vaccine product through phase 1 testing in the US and also phase 2 testing in Korea, and we're through our support with CEPI are expanding that to a phase 2 trial in the Middle East and other appropriate geographical regions starting, well, meant to be starting in very soon, as I'm sure many of you on this call can understand. Unfortunately, there has been, of course, impact of COVID-19 on ongoing clinical trials, but so far we're running on track to start that phase 2 in the Middle East and Africa very, very soon, certainly in the next few months. So that's ongoing. So yeah, we're moving, of course, in the right direction for our MERS candidate. We're very excited by the data that we're seeing. One thing I think we thought was very important, and I think that was confirmed by other published, was the importance of T-cell responses for protection in convalescent patients, and that was something that we were able to really robustly generate with our INO 4700. I'm really sorry, we're very close here in San Diego to the Air Force Base, and there's fighter jets going over the top of me, so I apologise for that. Yeah, we were able to demonstrate very strong T-cell responses, and something that was particularly heartening to us was we were able to get access to MERS convalescent sera from the outbreak in Korea, and so we were able to run that convalescent sera next to our vaccine-induced clinical samples as well. And in every case that we tested on the neutralizing antibodies, the binding antibodies, and the T-cell responses, we were extremely similar to the levels that we saw in those convalescent patients. So we feel that's particularly strong supporting data for the platform for coronaviruses in general.
Fantastic. I noticed we're at 12:02, which is beyond the original ending time, but of course we will continue a little bit longer. But I did, for those who have to leave us, wanted to point out that there are 2 additional talks about other vaccine strategies. One of them tomorrow will be Dr. Greg Glenn from Novavax, and the other on Thursday will be Dr. Tal Zaks from Moderna. Novavax is developing a protein-based vaccine produced by baculovirus, and Moderna an RNA vaccine. So it's an opportunity to hear about additional vaccine technologies. So there were a number of questions about funding and the collaborations that it takes to put all of this together. And one of our questioners said, compared with what they've read other companies have gotten, the amount of money you've received from CEPI has been relatively small. And others have questioned whether, or asked the question of how are you protecting your intellectual property through these collaborations, including with countries where some concerns have been raised. And so, can you talk a little bit about the funding and collaboration aspect of the project?
Yeah, absolutely. And firstly, I would like to reiterate how incredibly grateful we are to all of our supporters, including those who I spoke, CEPI, the Gates Foundation, the Department of Defense, all of whom have been incredibly generous to us. We have had, I think the CEPI partnership was perfectly positioned because we already had a very large existing relationship with them. They'd given us $56 million for our MERS and LASA program. So I think the follow-on funding for COVID made a lot of sense there. They were very familiar with our technology. And as of yesterday, I think we were their biggest, we were the partners that they'd given their the largest amount to. I can't obviously talk about things that aren't publicly announced at the moment, but we're in discussions with other funders moving this forward. And as I'm sure everyone on this call can imagine, vaccine development under normal circumstances is expensive. Try doing that under rapid circumstances makes it even more so. So really, the support that we and of course all of our colleagues working in this area have got has really been so instrumental to allowing us all to push forward these things, these solutions, these vaccines, these therapies as fast as possible. So we're very grateful to that. On the intellectual property side, yes, you can imagine that is a major concern, and we certainly have been very very cognizant of that, I think, is maybe what I would like to say as we move forward with our partnerships.
Great, thank you, Kate. A question here about electroporation in general, which at least as you read the name, sounds like it's scary and might be painful. So can you say a little bit about the acceptability of that in patients?
Yes, absolutely. And I think this comes back a little bit, Phil, to I think your opening question, you know, and it's completely understandable. I think, you know, people maybe have a historical view of what DNA vaccines are and what electroporation was. But really, you know, I would want to reiterate that things have changed really quite considerably in the last decade or so. And I think what perhaps the historical view of both of those things really isn't what they are today. So certainly let's talk specifically about the intradermal electroporation that we're using for COVID-19 here. As I mentioned, the device itself is about the size of an electric toothbrush, battery operated, handheld, super easy to use. You know, we designed it specifically for ease of use by the clinician. But equally as important to that, we also designed it to be as efficacious, of course, as possible, in that it's delivering as much DNA as possible, but also that it's as acceptable to the patient. And we've published actually that intradermal electroporation on a visual analog pain score is considered exactly the same as what it is for a standard IM injection, so your standard flu shot. So from a perspective of a patient, the processes would feel exactly the same. There's really no difference. And the electroporation itself, it's extremely rapid. The pulses are sort of 52 milliseconds, so, you know, less than kind of a blink of an eye. So certainly from the perspective of a patient's tolerability, their experience of the whole vaccination process, it would feel no different to them than their standard flu shot. And really that's the feedback that we've got consistently using our intradermal delivery technology, and certainly the feedback that we've got from the patients enrolled in our COVID-19 study.
Okay, and just a few more fairly technical questions about the immunology and the expression. One of them, obviously the vaccine induces both humoral and cellular immune responses. Can you say a little bit about the parts of the virus life cycle then as it infects a human that the vaccine-induced immune response is likely to be able to prevent or attack?
Yeah, absolutely. We feel that the ACE blocking work really is very confirmatory that we'll be able to, or potentially able to, our vaccine will potentially be able to block the binding of the spike protein to the receptor on the cell. So hopefully being able to stop that process before it even starts. So we feel quite confident that that's the mechanism of action, of course, but there's a lot more work and analyses to be done. You know, it's still hard for me to wrap my head around that, you know, we've only known about this virus for 5 short months. So there's still a lot we have to learn about it, but we're hopeful that that's how the mechanism of action of our vaccine.
Good. And the spike protein as it's presented, do you know if that forms trimers or is it perhaps presented as monomers?
A great question. I'll be completely honest and say I don't know the answer to that, I'm afraid. If anybody else on does know, I would love to know, but no, I'm sorry, I can't answer that.
And here another very technical question from somebody who's very carefully read one of your papers, which is that your plasma design includes IgE leader sequences. And the question is, what is the role of such sequences in immunogenicity or efficacy?
Yeah, absolutely. Great question. And actually, our IgE leader sequence is used routinely for our vaccines. It just really helps shuttle the DNA to the correct part of the cell. We have done a lot of, as you can imagine, and I'm sorry those jets are going overhead again here. I'm sorry, that's happens about 10 times a day. that really, we've of course done many head-to-head comparisons of that, and have shown that the leader sequence really does enhance immunogenicity, and so that's why it's included in our vaccine constructs.
Okay, fantastic. Well, that actually, I think, covers, at least in a broad sense, essentially all of the questions that we have. Well, actually, there was one more question, but I think you answered it just about the preclinical information you had before going into humans. Somebody asked how much animal data was required. And I think what you mentioned was that because of a lot of experience with the vector, you were able to move into human studies with relatively less animal data than other products would have.
Yeah, for our filing of our IND, we used the mice and the guinea pig data.
Okay, well, good. In that case, what I will do is thank you very, very much, Kate. We really appreciate the very clear talk, and especially appreciate you staying over to answer some of these additional questions. Very clear answers to these questions as well. I have to say you have an exciting life, and you're in the middle of something that is really important. And so keep up the good work, and I think we all wish you success.
Thank you so much.
Great. Wing, are there a few words you would like to say as we close?
I just really thank everyone for your attention. Obviously, thank you, Phil, and thank you, Kate, for helping deliver such an interesting session. Apologies that there were so many questions that we weren't able to answer them all, but hopefully there'll be another opportunity to do that. And also a final thank you to our webinar sponsor, Sino for today's webinar as well. Just to remind everyone that this session, this video is recorded, and the link is due to go out tomorrow. I may do a little bit editing just to get that original technical editing out, but otherwise I hope you can all tune in tomorrow, as Phil has mentioned, for Novavax and tomorrow, and Moderna for the day after, and all of those details and how to register can be found on our World Vaccine Congress website.
Well, thanks again, and look forward to seeing at least you show up as the number of attendees tomorrow and on Thursday. And thanks again, Kate.
Thank you.
Thank you, everyone.